Welcome, everyone. Before we start, we wanted to say thank you for your participation today. And today, we are going to kick it off, as we always do, with the good Dr. Anthony Schmidt. He is our Senior VP of Science. He means a lot to Arima. He's been the foundational scientist behind our platform. He's been the lead developer of a number of technologies, namely Arima-HiC chip and Capture-HiC. He also has a lot up his sleeve, as always, for new product launches for later this year, which you'll have to stay tuned in future webinars to learn more about. Anthony, wanted to just introduce you, say thank you for all you do for Arima, and give you the floor. Floor yours. Cheers.
All right, so welcome everybody. Thank you for attending. I'm excited to give this technology introduction in advance of Dr. Schneiderl's presentation on how he's used the Arima-HiC platform to uncover insights on undiagnosable tumors. Really, there's just a four-part agenda for my introduction today. I want to start by just describing what is 3D genomics, or what do we mean by 3D genomics, provide an introduction to the Arima technology platform, and then go into some specifics on, or some sort of conceptual introduction on how we use the Arima technology to identify structural variants such as gene fusions, and then conclude with just a quick benchmarking study summary where we've used Arima-HiC technology to identify structural variants in FFPE tumors that had been previously characterized by orthogonal methods like FISH or targeted DNA or RNA panels. So just a little bit about Arima. Our sort of goal here is that we're advancing the life sciences through the power of 3D genomics. And we'll talk about what we mean by that. And we do that by developing innovative solutions that drive discovery in genomics and improve human health. You'll see throughout the presentation that I'll talk about the Arima-HiC technology platform, and then how we provide access to that technology via kits and reagents, as well as and services. So anything that you see in my presentation, as well as the one from Dr. Schneiderl, can be done in your lab through the use of kits and bioinformatics tools that we provide, or sending your samples to us for an end-to-end service on sample prep with optional sequencing and informatics. So very broadly, how can 3D genomics empower cancer research? Really, this slide is meant to just convey sort of three key areas of application of this technology for biomarker and therapeutic development. First, the detection of known fusions as well as novel fusions. So we'll see some of that from my presentation as well as Dr. Schneiderl's. Finding gene fusions that are missed by other technologies, as well as the study of structural variants and gene fusions in retrospective or archived FFPE samples, things that are 5 or 10 or more years in terms of archival period. So if any of these sort of three key areas are directly relevant or tangentially relevant to sort of your research, then this 3D genomics approach may be of interest for you. So what is 3D genomics, and how can it reveal new biology in cancer? So at the highest level, to compare 3D genomics, we kind of juxtapose that to linear genomics, or 1D genomics, where we're all very well aware of the typical linear representation of the genome, a string of approximately 3 billion A's, T's, C's, and G's. And 3D genomics is similar to linear genomics in that it's a sequencing-based data type, so it still sort of uncovers the underlying sequence of the genome, but it also gives you a measurement of the three-dimensional structure of the genome, which we've tried to illustrate here in this sort of graphic. So you get a measurement of that three-dimensional structure of the genome through a sequencing data type that sort of also provides the sequence level information as well. And some of the exciting things that this data type enables is the ability to look beyond just the sequence level information of the genome, which is sort of typically how we understand genomes today. So in this sort of iceberg type of schematic with sequence level information on the top, the 3D genomics approach allows you to sort of look deeper into other application areas like the detection and characterization of structural variants such as translocations or inversions. And then beyond that, provides really valuable insight into the study of how oncogenes are regulated through three-dimensional gene regulatory actions, such as those between enhancers and promoters, and even beyond that, just some fundamental questions about how the genome is organized and how that relates to genome function. And the majority of what I'll talk about today is really going to be focused on this structural variant piece. But I did want to maybe just start with one very quick vignette on the relevance to gene regulation cancer research, and then I'll flip back to structural variants for the rest of the presentation. And I wanted to just highlight this really interesting study that was published a couple years back now from the Iafontis and the Syragos labs. This is all published, so you can look at this yourself. But we've kind of rolled this up into a schematic that highlights some of the advantages of using this 3D genomics approach to understand disease mechanisms in T-cell acute lymphoblastic leukemia. And what's shown here is that if you 3D genome profile healthy T cells from healthy individuals, what you know here is that here is the MYC oncogene, and then some 2 million base pairs away is a putative enhancer element that in healthy T cells is not active. And by obtaining that three-dimensional view of the genome, you actually can find that the MYC oncogene, which is sort of in green here, is spatially segregated from this enhancer element in healthy T cells. So it gives some clues or evidence as to why that enhancer element is not regulating MYC in the context of the healthy state. However, if you do this 3D genome profiling of T-cell acute lymphoblastic leukemia patients, well, MYC expression is actually activated, and some 2 million base pairs away, there's transcription factor binding or activity at this enhancer element. And it would be very hard with a purely linear view of the genome to attribute this type of enhancer activity to have a regulatory effect on MYC about 2 million base pairs away. But with the 3D genome profiling view, we actually saw that these two neighboring domains up here coalesce or sort of merge together, such that now this enhancer element bound by this TF can come into that three-dimensional proximity with MYC to activate its expression. What's also really interesting about this is when you have that view that this transcription factor is binding to this enhancer, which is regulating MYC, the authors of this study could then say, well, if there's a small molecule available to target this transcription factor, then that might have an effect on how MYC is regulated. And that's exactly what they did using a CDK7 inhibitor. And so when you treat the cells with that CDK7 inhibitor, MYC expression goes down and ultimately the cells end up dying. And the enhancer element no longer contacts MYC. So really this just sort of underscores the role that 3D genomics can play in identifying the sort of oncogenic gene regulation, and helps you inform which therapeutic strategy might be effective based on that 3D genome structure. All right, so flipping back to the structural variant part of the story, I probably don't have to say too much about this, but SVs have an incredibly significant role in cancer. I think that's fairly well established, with more than 95% of cancers have one or more somatic structural variant, and at least 30% of those cancers have a known pathogenic structural variant that's used in the diagnosis or patient stratification. What's probably less known, although there's some very valuable references here at the bottom from the labs of Wang Wei and Jesse Dixon and Lucas Chavez, and others have also seen this as well. But really, SVs can impact the regulation of cancer genes through three-dimensional interactions that occur at the breakpoint or as a result of a structural variant. Some very interesting research along those lines as well. But ultimately, the detection of SVs is essential for elucidating disease mechanisms. And we also appreciate that understanding SVs, such as the detection and the subsequent characterization of the disease mechanisms, of course, requires an integrative approach. And we feel that 3D genomics is a valuable tool both in the detection and the characterization of those structural variants, but it really requires an integrative approach for a complete understanding of SVs in cancer. I just wanted to show this one figure from the PCOG study published back in 2020. I won't go into any of the details, but just to highlight one of the observations from that paper, which was that cancer genomes contain, on average, 4 to 5 driver mutations, and those mutations can be both in coding and non-coding regions, and not all cancers have detectable driver alterations. It really underscores, I think, the importance of having a tool like 3D genomics that can look both into the coding and the non-coding regions and identify both known and unknown drivers of cancer. And so ultimately that kind of rolls up to this slide, which is, how is 3D genomics, powerful for cancer research? And I've already sort of touched on the ability of it to identify structural variants like gene fusions, detect that 3-dimensional interaction like in the Iafontis and the Sirigo study, to yield insights onto how oncogenes are regulated, and then still not forgetting that the data type provides that underlying sequence information because it's a NGS-based assay sequenced on NGS platforms of today. All right, so a bit about Arima technology. I first want to start with the end-to-end Arima-HiC workflow. So for this 3D genome analysis, the actual technology that we used is called HiC, or Arima-HiC. That workflow starts with sample prep using the Arima-HiC plus kits and reagents. And if you zoom in on just what the Arima-HiC plus workflow is that comes at the very beginning, well, we take a variety of different sample types or input quantities like blood, cell lines, frozen tissues, as well as FFPE tissues that I'll spend most of the time talking about today. And really what we do is we take advantage of the fact that within all of those cells from all of those sample types, the genome is folded into this 3-dimensional structure. And if you zoom in on just this blue chromosome here, parts of that chromosome are going to be held together in 3-dimensional space. And all we do, like if you take this light blue piece here and this dark blue piece here that are spatially proximal, all we're doing is cutting the genome and then ligating it back together so that this light blue piece and this dark blue piece that were spatially proximal are now physically linked together. And we ultimately want to sequence those molecules that are physically linked together so that we can learn something about which 2 pieces of DNA were spatially proximal in the genome, right? So that's a measurement or a data point for the 3D genome. So really after the Arima-HiC process, what comes out of that is just purified DNA that has been HiC'd, so to speak, where the spatial proximity information has been preserved in the DNA molecules that gets output from HiC. And that just flows into standard pre-validated Illumina library preparation protocols, short read sequencing, and then on the back end, the Arima SV pipeline to identify the SVs from this HiC data. And what our customers appreciate about this workflow is that it plugs into the existing NGS ecosystem. So if you're already running some sort of DNA sequencing assay or RNA sequencing assay, the only difference really to this whole end-to-end workflow is a different DNA extraction kit from us that does this HiC method and outputs DNA ready for library preparation. And then on the back end, we have containerized bioinformatics solutions in Docker or Singularity that provide portable and scalable analyses. And sort of altogether, we feel that this provides a pretty robust and reliable pipeline to identify SVs, and we'll talk more about that in this presentation in Dr. Smundaro's. So next, I want to conceptually go over how to use this HiC data type to detect gene fusions. And the first thing I think is really important to understanding how the technology works to detect gene fusions is to understand what happens within in a karyotypically normal cell. So what's shown here on the left is within one cell, you have chromosome B floating around in its own space, chromosome A floating around, occupying its own space. And these little dark dots here represent those contact points between different parts of chromosome B, as well as different parts of chromosome A. So those are those spatially proximal parts that will be cut and ligated back together in Hi-C. And when you transform what's happening in vivo to sort of a read structure along the linear genome, here within chromosome A on the left, you'll have these interaction points between two different parts of chromosome A, right? So from here to here, and underlying is that paired end read structure where you have read one on this part of that three-dimensional linkage, and then this read two from that three-dimensional linkage, right? Representing that one interaction point. And then on chromosome B, you also have the same thing. Here, there's three different interaction points within that one cell. And the key takeaway here is you get these 3D linkages within chromosomes, and you very rarely, or in this case, not at all, get any types of three-dimensional linkages between chromosomes. So that's sort of the basal state in healthy cells. Now, if you flip that and you have a scenario where you have chromosome A here in light blue and chromosome B in dark blue, and there's been some sort of a translocation or rearrangement of sorts, and it creates a breakpoint here where those two chromosomes have fused together, right? So now that's the fused chromosome within that cell, and you still have lots of 3D linkages within that fused chromosome. And what that looks like, sort of in a linear representation, is imagine you have a gene fusion here between gene A and gene B, right at that breakpoint. Well, you'll have lots of these reads that are three-dimensional linkages between chromosome A around gene A to chromosome B around gene B. And the presence of those have a very low background rate in healthy cells, but they're omnipresent in cells that have this particular gene fusion. So this might be one cell. If I turn to another cell that has a slightly different three-dimensional configuration around that gene fusion, you'll get many more of these three-dimensional linkages that link the gene A region to the gene B region. And you can imagine going through cell 3 with different 3D linkages, and ultimately here cell N, let's say this is cell 60, where you have two things that I really wanted to highlight here. Is what's really obvious is you have all this mass amount of linkages between gene A and gene B that provides a sort of this amplified signal that there must be some sort of gene fusion happening here, because you wouldn't expect that many links between gene A and gene B. But also, if you sort of take your eyes here and you scan down, it's also important to note that there will be reads that are at the breakpoint, right? Which is right here. So if you scan down, in this case, there's about 6 reads that are actually at the breakpoint to provide that breakpoint level evidence, but there's also about 10 times as much signal linking gene A to gene B, sort of amplifying the evidence of some type of gene fusion or rearrangement happening in this particular sample. And that's really one of the advantages of this 3D genomics approach, is retaining that sequence level information at the breakpoint, but also getting that 3-dimensional long-range interactions that link the two chromosomes together. Now, what does some actual data look like? We often depict this 3D genomics Hi-C data as a heat map. And just quickly to orient you with what you're looking at here, this is a genome-wide Hi-C heat map that was recently published as part of the first human T2T genome assembly paper, where they used Arima-HiC as a means to help construct that T2T genome. This is a karyotypically normal sample where the one thing that sort of really stands out is if you plot all the chromosomes along the this axis here from 1 to 21, here they're ordered by size. And again, from 1 to 21 down here on the bottom, one of those Hi-C linkages that, let's say, links chromosome 1 to somewhere else on chromosome 1 would be one single red data point on this Hi-C heat map. Right? And so you can amass these types of heat maps from sequencing many reads and many cells. And one of the things that I should also mention here is that the more red you see, the more three-dimensional linkages there are between any 2 points along the genome. So what stands out like a sore thumb here is that there's lots of three-dimensional linkages within chromosomes, like I showed you in the schematic. So there's lots of that three-dimensional folding within chromosomes, and relatively little three-dimensional interactions between chromosomes, like I showed in the schematic. And this is just some actual data to support that schematic. Right? So that's a really important point about these genome-wide Hi-C heat maps, is lots of intrachromosomal linkages and very few interchromosomal linkages in a karyotypically normal sample. Now, I were to zoom in on, let's say, a 100 kb region around within chromosome 1, there's other important features of the genome that are relevant for how we use Hi-C to detect structural variants. And one of the sort of foundational properties of Hi-C data is any two regions along the linear genome that are close together are also spatially proximal. So if two pieces of the genome are 5 kb apart, then they will also have the highest number of 3D linkages. Okay, and as you sort of move further away, let's say you have a region on the genome that's at position 5K and position 35K, the number of 3D linkages between those will be less than when you have two pieces on the genome that are very close together. So, and as you continue to move further and further away along the linear genome, there's less three-dimensional linkages. I think the main take home here is that you can tell from Hi-C data that when things are spatially proximal to each other in three-dimensional space, they're also linearly proximal. Okay? And that turns out to be really useful, as you can imagine, to detect gene fusions, because those two fusion genes that are now linearly right next to each other are also going to show really high spatial proximity. And so here's a schematic of chromosomes 2 and 5. Chromosomes 2 and 5 are right here in blue, 2 and 5 in this cartoon Hi-C heat map. And you have a typical high intrachromosomal signal. And then you can ask yourself, if you have a reciprocal translocation between chromosomes 2 and 5, what type of Hi-C pattern might you expect looking at that Hi-C heat map? Well, what you would expect is a very high number of these 3D linkages between chromosome 2 linked over to chromosome 5 and vice versa. And that'll look something like this. So if you look at the chromosome 2 interacting with chromosome 5, you'll see this really strong intrachromosomal strength signal between chromosome 2 and 5, which ultimately clues you in that there must be some type of event going on there. And ultimately, that's what the algorithms that we use ultimately try to harvest to identify structural variants and gene fusions in these cancer genomes. One very brief point that I wanted to make, but I don't have time to go into it in detail, is that different patterns in that Hi-C heat map will actually tell you something about the specific nature of that structural variant. And for example, if you have a interchromosomal reciprocal translocation, it tends to make these bow tie or butterfly-like patterns in the Hi-C heat map. If you have just a translocation where one segment, like here in A, goes over to chromosome B, you'll just have this type of pattern. And intrachromosomal events like deletions and inversions also produce their own signatures, which is useful for detection of a particular breakpoint, but also clues you into what type of SV there might be in play there. Okay, so shown here on the left is some actual Hi-C data from a karyotypically normal sample. And shown here on the right is a colorectal cancer sample that had a ROS1 SLC34A2 fusion. So you can see in this sample that there's actually lots of these off-diagonal 3D genome linkages that link different chromosomes together at a really high signal strength, right? And those would be the evidence for structural variants. If you zoom in to just one of these, I kind of wanted to walk you through how these data are interpreted. So here I'm zooming in at a pretty high resolution, like the couple hundred kilobases around this structural variant. And if I drop in the genes at this locus, as well as their orientations of the 5' and the 3' notations, here is ROS1, here's SLC34A2. And what this particular Hi-C heat map here shows you is where the breakpoints are between ROS1 and SLC34A2. And it turns out that they're right here, sort of at those purple lines. So where the purple line intersects ROS1, and where this purple line on chromosome 4 intersects SLC34A2. So how does this heat map actually tell you where the breakpoints are? Well, think about it, if you just focus on this lower right quadrant, this is essentially saying there's a lot of spatial proximity signal and 3D genome linkages between the 5' side of ROS1 and the 3' end of SLC34A2. Okay? And so what that actually represents when you think about it as a schematic is lots of these 3D genome linkages from the 5' side of ROS1 to the 3' side of SLC34A2, and that clues you in on the structure of this particular gene fusion being a ROS1 SLC34A2 fusion. But because this is reciprocal, you also have this really high spatial proximity signal between the 5' of SLC34A2 and the 3' end of ROS1. So the reciprocal chromosome structure here is the SLC34A2 to ROS1 fusion in this particular gene structure. So lastly, I just want to wrap up after sort of introducing the Hi-C data type and how it's visualized with a benchmarking study that we've conducted with 3 institutes. 2 of those here are Children's Mercy Kansas City, as well as Scripps MD Anderson, consisting of 24 samples that had been previously characterized by FISH or targeted DNA or RNA panels. This spanned 19 different tumor types and somewhere between 1 to 12 years of archival period, depending on the particular sample. And on average, about 300 nanograms of DNA recovered from these samples on average, even though the spread was less than 10 up to over a microgram. And in the first 12 samples with Scripps MD Anderson, here are the 12 tumor types listed here on the left. Here are the results from the Arima-HiC analysis showing the fusion detected. You can tell that many of these were actually ALK fusions in lung tumors, although we do have bile duct tumors with FGFR fusions, as well as a breast MASC tumor here with an NTRK fusion. And all of these, like I said before, were characterized with some orthogonal assay with some known fusion shown in this column. I just wanted to highlight this one breast MASC tumor with the NTRK fusion because it's a pretty interesting case where if you look at this genome-wide Hi-C heat map and you zoom in on the chromosome 12 by chromosome 15, you see lots and lots of these breakpoints between this part of chromosome 12 and then the tail end of chromosome 15, like some type of chromoplexy event. And within these complex rearrangements, if you zoom in on just one of those breakpoints, what you see here is this 5' end of ETV6, highly interactive with just the 3' end of NTRK3, suggesting this ETV6 NTRK3 fusion, like you see here in the schematic. And lastly, the work with Children's Mercy Hospital. We have 5 different tumors here, all FFPE tumors, like the Scripps collaboration, somewhere between 8 and 12 years of archival period. These happen to all be FOXO1 PAX fusions in alveolar rhabdomyosarcoma that we can detect as well with 100% concordance between us and the orthogonal method. So just to wrap things up, if your research is related to these things that we sort of talked about in this introduction, then 3D genomics might be approached for you to detect clinically relevant gene fusions in a host of different sample types, including FFPE samples and archived FFPE specimens. To learn more, you can go to our website. And then I just also wanted to, again, plug the opportunity for the free kit if you apply to our cancer research grant. Thank you very much.
Perfect. Thanks, Anthony. Always appreciate the insights from Arima, pulling the sheets back a little bit on things that you guys have been up to in the lab. And for further detail, wanted to introduce Dr. Matthias Studerol. He is a neuropathologist at NYU Langone, whose clinical research focuses on identifying novel biomarkers for diagnosis and therapy. Dr. Studerol's laboratory studies cancer genetics and epigenetics and examines the molecular differences between tumors at the cellular level. Matthias, good to see you since AACR. Hope all's been well. The floor is yours.
Thank you so much, Christian. Can you hear me well?
Yes.
Fantastic. And I would like to thank Arima for the opportunity to share some results of our work in this exciting topic. Let me just share my screen. Okay, so let me tell you a little bit more about how we are using the HiC approach to unravel the complex nature of the genetic origins of brain tumors using this 3D genomics approach. And it's really great to follow up Anthony, who gave such a great introduction into a technical aspect, which I'm not the master of. So the first question I often get is, why on earth do we need more molecular tests? So just to give you a flavor of what we do at NYU, we have a very extensive molecular pathology portfolio. We have a DNA sequencing panel that is currently the largest in the country and FDA approved with 607 genes. It's a matched tumor normal. We have a fusion seeker panel with which is an RNA sequencing assay for 120 something genes. We do a whole genome DNA methylation profiling of all the cancers. And then we have a bunch of smaller 50 gene targeted panels, both for solid and hematological malignancies and various targeted assays. So why do we need more? Well, it's because that promise of precision medicine still remains somewhat elusive. And I don't think I need to explain to anybody in this audience the idea of, rather than going strictly by a histological pathology, the idea of precision medicine is to identify specific genetic drivers, and then chemists will develop wonderful small molecules that will inhibit the tumors. But it doesn't really work that way. So I just want to show you one of the cases that we've been working on. It's a 15-year-old female with NF1 who presented with new onset of headaches and seizures. Subtotal resection was performed. The diagnosis was relatively descriptive, calling it a low-grade glioneuronal tumor, and recommended was just a follow-up with monitoring. We did our molecular profiling, and you can see here on the genome pack, we just see a spliced donor variant in p53 and a couple of passenger mutations of unknown significance. It was negative for gene fusions on the fusion seeker, and DNA methylation classified it as pilocytic astrocytoma methylation group family pilocytic astrocytoma with a classifier score that was somewhat unusual. And here you can see we can also utilize DNA methylation to pull out the copy number profiles. So I'm going to put this case on the back burner and return to it in a little bit. So what are current challenges in the identification of genetic drivers of cancer? So first, clinical panels, even as extensive as at NYU, still fail to identify drivers and therapeutic targets in many tumors. And some cancers, despite all the landscape genomic efforts, still lack molecular drivers. Clinical panels are predominantly focused on coding regions of the genome, and the cancer driver genes are often tissue context development dependent. There is also not enough high-quality frozen tissue for research, so the discovery that is limited to formalin-fixed paraffin-embedded tissue can be challenging because there is a significant degradation of nucleic acids, particularly RNA, and we know from our experience and others that RNA sequencing does not perform well on tissues that are more than 4 or 5 years old and have been embedded in FFPE tissue. So we are moving towards the 3D genomic exploration in cancer research. We are looking for drivers, mechanisms, and targets for therapy, and NYU Molecular Pathology in my lab have partnered with Arima-Genomics to develop a novel approach to characterize cancers with unknown drivers and therapeutic targets to identify a robust, clinically actionable high-C profiling. So let's go back to our patient who, after a resection, rapidly recurred in 6 months. This is MRI. You can see again the tumor here has recurred. On histology, looks much more aggressive, much more pleomorphic. It was diagnosed at this point as a glioblastoma, a malignant brain tumor, and negative for gene fusions. You can see the genome packed, identified all the mutations as we've seen before, but also numerous copy number gains and losses that are listed here in this box. When we compare the copy number profiles between the original primary tumor and the recurrence, you can see it is much busier. You can clearly see a CDKNA homozygous deletion here and multiple things happening on 9p. And when we zoom in here, you can see that the 9p chromosomal arm was normal in the original tumor, but it seems to be shattered with the gains and losses in 9p. So we perform, with the help of Arima-HiC analysis of this recurrent tumor, and we identify a rearrangement that involves a PD-L1 locus that actually sits on 9p in this pediatric glioblastoma. And you can see this nice zoom in as Anthony was describing the significance. But it was not a gene fusion in a classic sense. It was basically a rearrangement of PD-L1 with a non-coding part of the genome. And we didn't really know what to do with it. So we follow up with a staining for PD-L1 by immunohistochemistry, and you can see this is a very strong positive stain for PD-L1 in this tumor with a rearrangement. And just to give you an idea, is we don't really do PD-L1 staining in brain tumors because it's rarely, if ever, positive, so we don't really have any use for it. But this was very encouraging because it showed that the rearrangements that we saw in high-C actually has a functional consequences, and it resulted in a change of therapy. The patient is currently on immunotherapy, responding well and stable. And with this way, high-C was able to identify a cryptic rearrangement that involved PD-L1 and was not detected by traditional methods, and we were able to confirm it by immunohistochemistry. We are working on integrating high-C with a whole genome DNA methylation for a discovery of drivers in brain tumors. These are published data that was pioneered by Stefan Pfister's group and David Kapper in Heidelberg, and is basically a epigenetic map of all brain cancers using whole genome DNA methylation profiling. NYU was first in the country to be approved by New York State to perform it in the clinic, and it gives us a very good view on the landscape, on epigenetic landscape of brain tumors and other cancers. So how do we combine high-C and DNA methylation? So this is a good example as a case of a 37-year-old female who had new onset seizures and had a 2-centimeter non-enhancing temporal lobe mass, and she underwent a resection. On the diagnosis, it was diagnosed as a diffuse infiltrating glioma, and the DNA NGS was negative for IDH1 and 2 mutations, which we would sort of expect in this age group in diffuse infiltrating gliomas. And it was sent to us for DNA methylation profile and RNA sequencing to see if it's one of the pediatric type of low-grade gliomas. You can see when we perform DNA methylation profiling, it's classified with a very high confidence score as a low-grade glioma MYB subtype. Those are tumors that include both MYB and MYBL1 tumors, and it was positive by that, and you can see here clustered on TCE analysis very nicely with that group. The problem was in our fusion sequencing analysis, it was negative for any of the gene fusions. So this is a dilemma. What do you do when DNA methylation classifies as a specific subtype that is defined by gene fusions? And these are tumors that are mostly diagnosed in young patients with median age of 16 years. They are benign and they don't require any additional therapy usually. But without a diagnostic gene fusions, could this actually be a diffuse glioma that still requires adjuvant therapy? And how can we be sure that this is indeed MYB/MYBL1 driven tumor in absence of a fusion? And you can see here a completely flat copy number profile, not giving us any further indication. Using a Hi-C, we were able to identify MYBL1 mammal2 gene fusion. As you can see here is the zoom in. And I would just like to point out that this is all Hi-C performed on FFPE tissue, how incredibly clean the background is, and how easily, if you will, it is to identify this mammal2 MYBL fusion. You can see here 3 end of MYBL1 and 5 end of mammal2. So the implications are significant because we are able to resolve the discrepancy between fusion sequencing and methylation. We confirmed that it's a low-grade tumor of a MYBL1 subtype, recommended imaging and observation after the surgery, and the patient is not getting no additional adjuvant therapy. This is how the data look, as we've shown you before, as Anthony showed, these like overlapping reads that show us the fusion. But the major takeaway point for those who work with FFPE tissue in a clinical setting or in discovery like myself is that RNA sequencing may miss critical cancer drivers. And that could be due to various reasons. It could be because of the panel design when the partner is novel or uncommon. It could be RNA quantity, low cellularity lesions that don't produce a lot of transcripts that we can detect with sequencing. It could be RNA degradation that's particularly problematic for FFPE clinical samples. And we also observed that the low-grade tumors have a low level of fusion transcripts and therefore can make RNA sequencing also challenging to detect. How do we explore what I call the hidden landscape of complex rearrangements using Hi-C? This is another of our tumors with unusual histology. You can see this is a 20-year-old female with a new onset seizures, with a bright lesion in a right temporal lobe that we performed the resection, and you can see this very unusual glioneuronal tumor that was positive for synaptophysin. It was profiled by our genome pack. We didn't see any somatic mutations. We did observe a chromosome 7 gain, trisomy 7, and on brain methylation, it classified but very poorly as a dysembryoplastic neuroepithelial tumor with a calibrated score of 0.4. For those of you who are not used to DNA methylation profiling, just to give you some sort of a reference frame, we require a probability score to be higher than 0.9 to be confident that DNA methylation is accurately detecting the tumor type. And between 0.3 and 0.9 is this indeterminate area where we don't really know if the low score is because the tumor cells are diluted by normal cells, for example, inflammation, or is it because there's an unusual or novel driver that is skewing the score? So how do we resolve it? DNT subtype is usually driven by FGFR aberrations. Those are the most common drivers in these tumors, either mutations or rearrangements or copy number aberrations, but we didn't observe any of that. And these tumors usually don't have recurrent chromosomal alterations, and most cases have a flat copy number profile. But you see here that this tumor is a bit unusual, has a flat copy number profile with exception of chromosome 7. And when we take a close look on chromosome 7, and these data are derived from the DNA methylation, you can see there is a trisomy 7 that was also seen on next-generation sequencing, but you can see there are numerous breaks with the gains and losses throughout the chromosome 7, both on a P and a Q arm. When we performed fusion sequencing, to our large surprise, we observed that the tumor didn't have one driver fusion, but it had actually 3 unique driver fusions, including BRAF, ETV1, and EGFR, and the partner genes listed here. And what is interesting is all of these 6 genes involved in these 3 gene fusions are actually located on chromosome 7. So that alone is unusual, that you don't have one gene fusion to drive the tumor, but you actually have 3 unique gene fusions. So we could have called it a success and be done with it. We don't have one driver, we have 3 drivers, but we still did Hi-C because there were so many of these breaks and gains and losses. And when we perform Hi-C, to our huge surprise, we see this incredibly busy map. There is zoom in on all the fusions detected by Hi-C that we also detected before by RNA sequencing. But you can see that in addition to these 3 fusions that we detected by RNA sequencing, there is almost 100 unique events throughout the chromosome 7. So this is actually, I don't have a better word to describe it, but a complete chromotripsis of chromosome 7, isolated chromotripsis of chromosome 7. You can see these numerous events that we are literally digging through, trying to identify additional drivers. So HiC here uncovered massive shattering and rearrangements of the chromosome 7, which we now call isolated chromosome 7 chromotripsis. And you can see that inconclusive DNA methylation score often signals novel or uncommon drivers. RNA and next-generation sequencing identify really just a fraction, like we can call it a 3% of events happening on chromosome 7. And HiC provided us in-depth insight of genomic landscape of these rearrangements that we are now working through. So the next thing is, well, we can also use the HiC for rare cancers in archival tissue. And the problem with rare cancers are, as the name implies, they're rare. So there's not a lot of samples available for research, and a high quality frozen material is not often available. We can get a FFPE material, but that is not very good for RNA sequencing. So this is part of our program of molecular profiling of rare kidney cancers, which is a cohort of histologically unclassifiable kidney tumors on which we perform DNA and RNA next-generation sequencing. And we had this kidney tumor, it was called primitive neuroectodermal tumor that was diagnosed in 2012. There was no mutations on DNA NGS, and the RNA NGS failed. The blocks are more than 10 years old. But when we perform a HiC, we were able to identify a novel fusion between POF5F1 and TAF15. So you can see this is the gene that is involved in soft tissue tumors and carcinomas in gene fusions, and then TAF15 was previously described in gene fusions in acute mixed lineage leukemias. So you can see that the HiC performs really well even in 10-year and plus old FFPE samples. We're able to discover novel gene fusions and partners and really unlock ability to profile old and archival samples for research of rare cancers that we otherwise would be negative by standard DNA NGS and RNA NGS. In the interest of time, I'm not going to go into details in additional applications of 3D genomics and HiC. We can also identify neighborhood translocations, rearrangements between coding and non-coding rearrangements, and look into a chromatin architecture, loops and paths of cancers, which is part of our profiling effort. Example of that is this mesenchymal tumor of unknown driver that we identified translocations within 15 kb of TERT and MET, and we are working on validating findings that are coming through our pipeline to see whether these rearrangements lead to, for example, MET upregulation, and whether these could be potential targets for MET inhibitors such as crizotinib. And we have numerous findings like this that we are now validating with orthogonal methods. So HiC really adds diagnostic value in tumors with no detectable drivers. We have a pilot cohort of tumors with unknown drivers, recurrent progressive tumors, tumors that failed RNA NGS or had a poor quality DNA NGS or had an inconclusive whole genome DNA methylation result. And if we look at all the data all together, we concluded that HiC 3D genomics provides additional molecular information in 70% of cases. That could include potential therapeutic targets for currently approved therapies, identify novel genetic drivers, or some of which will require additional validation for sure. And we are also gaining insight into a 3D chromatin architecture. So we are significantly decreasing the number of cases that end up being negative for molecular drivers. To help those who do not have that capacity, we have decided to implement HiC discoveries in clinical practice and offer our ability to in partnering with other laboratories. So we have started what we call provisionally so for the NYU 3D Cancer Genome Hub, which is going to be an open access platform where collaborators can send us their clinical or research cases for profiling, paid only the cost of the reagents, and get back both NYU HiC and NYU DNA methylation results, as well as raw data, so they can integrate with their local in-house analysis, in addition to our pipeline outputs. Then we are hoping to serve, again, as we do with DNA methylation, as sort of an honest broker, connect the people who work on rare cancers and identify the rare cancer, and they can increase the cohorts, sharing unclassifiable cases to discover new tumor entities, and hopefully jointly confirming clinically actionable targets. So I welcome anybody who is interested to join this field to feel free to contact me. In conclusion, I hope I showed you that HiC provides really unique insight into 3D cancer genomic landscape. Our Arima-HiC protocol over our examination is robust and performs well on archival FFPE tissue in samples more than 10 years old, and HiC enables analysis of archival samples with failed or inconclusive molecular results. 3D genome analysis by HiC identifies novel molecular drivers and potential therapeutic targets, and we hope it's going to provide a future avenue for therapeutic discovery. I would like to thank a huge team of people that work with me and they make these things happen. And I would also like to thank Arima Genomics for their collaborative spirit and helping us with everything from profiling to interpretation of the data that has really powered this project. And that is everything. Thank you so much. Back to you, Christian.
Excellent. Well, thank you, Mattia. Really appreciate the work. Been great to work with you, meet you at AACR in person. I know Anthony's been working a lot with you recently. And anytime you can find novel rearrangements leading to positive therapy, I think our job is kind of done there, but you showed so much more impact of the technology, which is fantastic. We have a number of questions that have popped up, and I'll try to parse them out to the correct individual. You can definitely feel free to chat us if I'm getting that wrong. And maybe go back to give Mattia a break, give him a sip of coffee or water before we dive into some questions for him. I'll start at the top. A question coming in about 30 minutes ago from Arul. Thanks, Arul, for joining. What about intrachromosomal gene fusions? So gene fusions within the same chromosome. Anthony, can you address that question?
Yeah, they're absolutely detectable. I think in one of my slides, I showed examples of how deletions or inversions perturb the IC heat maps, and they are detectable by the algorithms that we use. And then also in some of the technology benchmarking we did with scripts, there were several samples that were intrachromosomal events like EML4-ALK, in one of them that we saw quite a bit, which is an intrachromosomal SP. And so yes, they are detectable if the breakpoints are on the same chromosome.
Perfect. Another question I think for you, I think it was just around the transition from your talk to Mattia's. Do smaller bits of DNA or RNA within cells get linked together?
No, really just double-stranded DNA to double-stranded DNA ligation. So it's not DNA to RNA.
Perfect. Can you speak to, and I think there's a couple of questions here at the end as well, so maybe we can just group those together. Just the sample input requirements. So thinking about FFPE, maybe fresh frozen tissue, scrolls versus slides, core punches, et cetera. What are your understandings of the variety of sample types that can be inputted and then kind of the amounts that you need to input into the assay?
Yeah, so for FFPE samples, you can start with unstained slides, 5 micron cut unstained slides. I think a lot of the work that we've done with Mattia has been on unstained slides. Another option is pre-cut scrolls, so 5 or 10 micron scrolls that are pre-cut into a tube. That also works. My internal process when we're analyzing samples at Arima is typically using about somewhere between 2 to 4 scrolls or unstained slides per sample. And in the vast majority of cases, that has been sufficient DNA for analysis. Occasionally, if it's some type of biopsy or there's low cellularity, then we'll use more than 4 sections, but my experience has been about 2 to 4 sections is typically enough. So that's for FFPE. And yeah, Christian, did that answer it, or was there any more?
There is an interesting kind of add-on to that question, just around the minimum amount of tumor cell representation. Do you or Mattia have any sort of guidance there? Thinking about percentage of tumor in the sample?
I don't think we've defined, because I think there is a question, and it's also a good point, is that the lower the tumor cell fraction or the allele fraction of a particular variant, the lower the signal is going to be, and the deeper that you'll have to sequence in order to see that over the sort of background Hi-C signal. I don't think we've defined that trade-off between allele fraction and sequencing depth precisely. And so
yeah, I can chime in there. That's one of our goals, is to actually establish the limit of detection. What is the tumor percentage that we need to be able to call? Just to give an example of the range, our genome pact has the limit of detection 10% of tumor cells for mutations, but our DNA methylation is 70% tumor cell content, right? So that's sort of the range. You know, DNA mutation sequencing can go really to small percentage, although you lose the copy number data in such a low percentage. So my gut feeling is the Hi-C LOD is going to be somewhere between 20 to 30%, just based on the data we've seen so far.
Okay, excellent. Let's see, we have another question from Arul. Thanks for the questions, we always appreciate it. We are gonna tick just a few minutes over the top of the hour, so I appreciate you guys sticking around and listening to the Q&A session. I think a question for Matiha. I didn't note the slide, but it was just about 10 minutes ago, just talking about the 3 driver gene fusions. Are they in frame?
I think when we did the RNA sequencing, they're all in frame, yes.
Okay. Okay, great. Let's see, a couple more questions just about FFPE processing. There's a question here, doesn't FFPE samples not have intact chromatin? Are there really any other steps needed to perform Hi-C from FFPE samples? And I think it's maybe thinking about the DNA extraction method.
Yeah. So conceptually, when we first started working on this, I thought perfect, because for a frozen non-FFPE sample, 2 of the first key steps are cross-linking the genome and then fragmenting the genome. Those are like the first approximately 2 steps for Hi-C analysis. And I thought perfect, because FFPE samples are already formalin fixed, and you're right, they are already fragmented. I think it's very well known that the DNA within FFPE samples, or the chromatin rather, is fragmented, but the 3D architecture is still very much intact. We've compared actually matched FFPE frozens from the same tumor and show very high correlation in the overall 3D genome signal. And I believe that's in our app note on our website.
Okay, great. Next question here is, actually referencing the recent publication of the T2T genome. Is there any established bioinformatic pipeline to process Hi-C data using this genome?
Yeah, that's a good point. Our SV pipeline currently is based on HD38. We have not made the CHM13 T2T genome as the reference for the SV pipeline yet. That's a great idea, and I can see us doing that in the near future.
Definitely. Let's see, a couple more questions and we'll wrap it up. Do you see a difference between sample types assayed? Maybe that's a question for both of you, actually. Be something that I can do.
I mean, just in terms of tissues, we've done a variety of tissues, so we are pretty confident. I'm not sure if that's what the question is, but we've done a variety of tissues, from brain tumors to sarcomas to kidney tumors. I forgot, we've been really testing it all out in various types of tumors, and there's no difference in tissue substrate. It can really on any tissue. The one thing, obviously, it's obvious, but it cannot be decalcified tissue, because that ruins the nucleic acids altogether. So the bone tumors that are decalcified, obviously we would never process them, but every other tissue that is a standard clinical fixation, it's acceptable for the protocol.
Excellent. And just maybe one last question for you, Matiha, that I was chatted. you showed, really good results in kidney and brain tumors. Are there other types of tumors where you'd really look to see this approach adding value?
Yeah, one of our biggest cohorts that we're actually powering through is sarcomas. So that's another soft tissue tumor, sarcomas. That we also saw very interesting data.
Excellent.
In the interest of time, we couldn't really show everything, but that's where we see, I mean, not surprising, it's the tumors that are driven by rearrangements, but that's where we also have some very interesting data that we are wrapping up.
Excellent. We are just 5 after the hour, so we should probably wrap it up here. Just wanted to, again, say thank you to you, Matiha. Great to see you at AACR. Thank you for your presentation there. Really looking forward to working together to expand your hub and more information to come there. And Anthony, it's always a pleasure to gain some insights into the inner working of Arima. Appreciate everyone's time. Thank you for joining. Thank you for your questions. You all have a good day. Cheers.